Gary Cook here with Dr. Kendra Furber from the University of Northern British Columbia. And Kendra is with the Northern Medical Program in Prince George. And she has conducted a lot of research, and I guess it's in Myelination. How do you say that?
Myelination.
Myelination. And it's neurological, and it deals when people have medical issues and they get older, from the communication from their brain down to their arms and legs. So, Kendra, does that sound correct or close?
Yes.
Okay.
So my research focuses on the cells that myelinate your nerve projections. So the nerves carry all the electrical signals that your brain is telling your body what to do. And so this myelin, these cells wrap this fatty insulator around those nerve cells, and then that allows that electrical signal to travel faster. So if we lose the myelin, either when we're aging or in disease, then that transmission is slowed. Our brain can't communicate with our muscles, or our different areas of our brain can't communicate as well for things like cognition and memory.
Okay. Is this like, I've got my arm is not quite what it used to be, and my nerves are there, my fingers are there, but it's not quite firing. Could that be one of the concepts you're looking at?
Yes, so it really depends on a lot of factors. So of course your nerve cells are affected, and then there's a whole other host of cells in your brain as well. So it depends on the area that the damage might have occurred.
Okay.
If this occurs kind of deeper in the brain, so your cell bodies kind of are on the outside, and then they project down into the brain, and then they'll relay that message down the spinal cord. So if the damage is in the areas that have not necessarily cell bodies, but those nerve projections where the myelin is, it can definitely contribute to symptoms that people might be experiencing.
Okay, we're going to come back to this more because it's quite interesting. So, why did you choose Prince George to the Northern Medical Program to conduct your research?
So, I've always had an interest at working at a smaller institution. One, you have smaller class sizes, you have smaller research labs, so you really have more of that hands-on experience with the students, and I'm able to still be in the lab. The University of Northern British Columbia has excellent reputation as a small institution, and as kind of a research-intensive small institution. And of course, the connections to the Northern Medical Program through UBC has other advantages for research collaboration and other research endeavors.
Okay. You've got some awards, like the Michael Smith Award for studying. How is your research in a practical sense? We're looking to say we'll use stroke survivors who, having some limbs and some cognitive issues and some mobility issues. How does that research affect them?
So yes, the Michael Smith Foundation has supported my Scholar Award, which is to build my research program and support research trainees to work with me. So we do a lot of research just in the lab, and we actually work with these cells in dishes and look at kind of how they function. And I know that seems really remote sometimes from the healthcare delivery and what people are experiencing when they have trauma or stroke or a neurodegenerative condition. But this has been my passion, because we don't understand how the brain functions. So if we don't understand that, it's really hard to figure out what went wrong or how to fix it. So as my research grows, I hope I can build more collaborations to kind of translate what we're trying to learn about these cells into disease models and maybe even into some sort of new therapies to kind of combat that neurodegeneration.
Is the neurodegeneration, is that you find it in when you get older and you're finding it with medical issues or diseases, am I correct?
Yeah.
Is, I guess you always have a hope, which you hope, I hope my research does this or hope my research does that. Is it too preliminary to look at your hypothesis now or your ideas, or are you just still trying to learn?
Yeah, so we just learn really a lot about these cells and how they kind of develop to form this myelin. So what happens is your brain, the adult brain actually has these progenitor cells for the myelin. And they can respond to injury and then remyelinate, but that doesn't happen very effectively sometimes. And if you don't get that effective remyelination, those cells are supporting the neurons. So if the neurons don't have the support they need, then they will eventually have more and more deficits themselves, or can die themselves. So yes, we are very focused on understanding how those cells develop, but hopefully this identifies some novel targets that can be used in therapeutics. So it is really, really very early, but this is kind of where the new ideas come from, and having new strategies to solve these really complex problems.
Okay. You've explained it very nicely. And I'm thinking like, oh yeah, from here down to here, and what the hell, why is it not working here? And it's I guess in one respect, it makes sense from a medical perspective, and from a perspective of a stroke survivor. Like, why is not my arm doing well, or why is my leg being longer than it should be? And I have this ideas here. I'm trying to focus it down there, but it's not. It's not listening. The arm's not listening. And what you've said makes a lot of sense, right? It's not the nerves itself, but it's almost like a coding. Am I correct?
Right. And so I don't want to, take away from all the important research. Of course, you get nerve damage, and the nerve cells are so important, but there's all these other cells in the brain that do so much, and everything has to work together as kind of a unit for things to function properly. So there's all these different aspects that we can kind of focus on to have different ideas or different strategies that might work in one circumstances, or for one individual better than maybe for another individual, depending, again, where the area of the damage is, even their age, and a whole host of factors can play into what's happening in the brain.
Is it tell me about the age factor?
Yeah, so just like your skin, when you cut yourself, my kid can fall on his bike, and in a day or two you can't even tell, and I do the same thing, and it's a week later and I'm still looking pretty banged up. So just like that, as you age, your brain has less, we call it plasticity. So it never heals completely the same way as it was in the beginning, but it has a lot of these adaptive mechanisms, and there are repair mechanisms that take place. But as you age, those mechanisms happen slower and slower, not as effectively, and not as fully as they would in a younger individual. So I think that's really important to consider with stroke or traumatic brain injury or other types of neurological disease, really what stage of life people might be at.
And it's so a 20-year-old with a stroke has a better chance, based on what you're looking at from the research, of recovering as opposed to somebody who's 50.
their brain will have a much more plasticity and be able to better adapt to that kind of new circumstances.
Okay. You mentioned before about the central nervous system, and part of your research, you looked at that. Tell me about that. Also, with the nerves and the myelination.
Yeah. So the central nervous system we define as our brain and our spinal cord, and then as our nerves leave the spinal cord out, to their fingertips, that's the peripheral nervous system.
Okay.
So they have different types of neurons. There's a lot of similarities, but they also have different types of cells that form this myelin. So again, there's a lot of similarities between the myelin and the peripheral system and the central nervous system. But the cells I focus on, called the oligodendrocytes, are just in the brain and the spinal cord.
Okay. Do those cells have a better chance of healing or regeneration as compared to the ones at maybe the end of the fingers or your toes?
You can see remyelination both in the peripheral and the central nervous system.
Yeah, but is it more prominent for your research for the central nervous system versus the peripheral one?
Well, I have a kind of a specific interest in cognition, so we're really interested in looking at those connections and tracks in the brain that connect maybe, where your visual system is to where your motor system is to where, you have your regulation of behavior, because you're making all these decisions and sometimes we have to make them really quickly. So that's just the area that I focus on more than the spinal cord or the peripheral nerves.
Okay. Some of the people who will be watching this have cognitive issues or stroke. Tell me, does that play an effect with your research?
Yeah, again, so if those tracks that connect those different areas of the brain are damaged, if those nerve projections are damaged or the myelin's damaged, that's when we can see it manifest as the deficits in cognitive function.
Okay, good. Now, everything you're doing is in the lab, right? In the petri dishes and all that stuff there. How do you do research? That is how you hope that you do right now that will expand in time.
Yeah, so my belief is kind of, again, understanding these basic functions of the cells. We can really have new ideas, because we have some really good strategies, and that's usually preventing secondary damage. But really to get that, I guess, full repair of the nervous system after an injury or stroke, I feel that we really need some new strategies. So we can look at cells in dishes. We have different models we can work up to that have more physiology, where we can look at maybe functional outcomes. But my expertise, again, is in the lab. So I'm looking to develop connections with other researchers and potentially even clinicians downstream that they can maybe use the knowledge that we're producing in the lab to affect the outcomes of people that have had a stroke.
Okay. You mentioned earlier, just a couple minutes ago, about secondary issues. Am I hitting that correctly?
Yeah.
Tell me, can you expand on that?
Yeah, so what happens anytime the brain has an injury, there's the injury site, but that cell damage or cell death, kind of, you can think of it as this spreading wave of destruction almost, right? And so a lot of what we do is trying to immediately reduce that impact, and there have been several strategies that have been very beneficial. But there are also kind of long-term changes, and we can still see there's some evidence that people, that have had an injury or stroke are more prone to kind of this neurodegeneration later in life. So if we can understand that better, maybe the delayed kind of effects, how to protect these cells or how to better repair the tissue that's been damaged, hopefully that will help, mitigate kind of some of the effects that come later in life as a consequence of that initial insult.
And some of the effects that come later in life could be everything from temper to loss of memory to confusion.
Yeah, I'm sure most of your listeners all have a much better understanding of that than I will.
They will, yes.
Yes.
There are many medical people who want to understand their stuff. Your research is looking at it's cross-functioning. So you look at Alzheimer's, Parkinson's, everything else that comes with that. Can you expand on that?
Yeah, so there has really been an interest in myelination in the past maybe 10 years or so. The amount of research has really taken off, both in the lab and clinically, looking at, so they can use really sophisticated imaging techniques now to look at the myelin in human brains, like if you guys are familiar with MRI imaging. So there's been a lot of work looking at correlations of changes in this myelin, or these white matter tracks where those nerve projections are running. In aging, you see a decrease in integrity of the myelin as our cells age. In many neurodegenerative diseases such as Alzheimer's and Parkinson's and Huntington's, they've shown effects. The myelin is affected. And then, of course, anytime there's an injury to those white matter tracks, such as stroke, the myelin will also be affected.
Okay. Wow. Now I'm thinking, like, where does this come from there? In the myelin is I said other people have hit it, and you're stroke survivors and Parkinson's and Huntington's and Alzheimer's. Is this myelin always under siege with normal people, or is it something that comes across when you have these degenerations in a neurological perspective?
Yeah, it is mostly prevalent neurodegenerative diseases. I should also mention multiple sclerosis, because that is the demyelinating disease where your immune system actually attacks your myelin.
Okay.
So I guess like any other parts of our body, those cells can get old with aging, and that's kind of just more of a natural phenomenon. But yes, mostly what we look at is related to different pathologies. These cells seem to be very vulnerable to stressors. The demand on them, because they actually will send out many kind of processes and form these sheaths on many different nerve projections. Yeah. So there's a high demand on them. So they're really vulnerable to that cellular stress. So if we can find ways to protect them or make them more resilient, or for them to, those immature cells that are around to better kind of develop and replace any of the myelin that's lost, kind of are the two strategies that we would have.
Okay. Two questions. The stressors, the cell stressors. Can you expand on that?
Yeah, so we did touch a little bit on secondary damage. So one of those things I said, there's this destruction of the cells, and they can actually dump their contents or send out signals that can stress all those cells around them. So that's kind of one of the stressors, would be signals from other cells or garbage dumped out from other dying cells at the injury site.
Do the cells repair themselves?
So the oligodendrocytes specifically, the ones that are myelinating, they have a very limited capacity to kind of repair themselves. They are generally lost, and then the new immature cells have to develop to reform the myelin that's there. So that's why, if we can understand how to push those immature cells to form that myelin when we need it, that's how we could stimulate the repair in injury.
And how does that happen now? Just slowly, sometimes not?
Yes. So actually, there seems to be some maybe individual factors with the cells themselves. So we look at lots of genes. So if the DNA is kind of the blueprint, and then it tells your cells to make all these building blocks that it needs. So it needs to make proteins, and it needs to make lipids and everything that forms that myelin. So we try to understand kind of that process, so we know where to intervene. So you do get some spontaneous repair, more so in younger individuals rather than older, but it's about the cells themselves, but it's also about the environment that the cells are in, right? So we said that the injury, all the cells are sending all these signals, stress signals, different kinds of signals, and those can kind of block the repair process. So if we can kind of, again, control that environment in the brain to make it more conducive for the cells to repair the myelin.
Is there anything, this is a long shot, is there anything a person who may be a stroke survivor can do to help that?
Well, again, I'm sure that you guys would have a lot of advice and resources that I don't have to give you, but in general, from other people's work, the same things that are healthy for your neurons are going to be healthy for your oligodendrocytes. So like a good diet and getting exercise and getting sleep and all those important things are also important for the cells that are myelinating.
If someone is stressing themselves and worried about something, does that also delay or inhibit the cells from myelinating too?
It can have impacts. Yeah, there's some research being done on that, different stressors, and there was a little bit of work done even before COVID on social isolation. So yeah, again, just like your nerve cells, all of your experiences and your environment can impact the function of the oligodendrocytes and the myelination.
Okay, good. Final thoughts.
Final thoughts. Well,
that's our concepts. Yeah?
Well, thank you so much for joining me, or having me join you, I guess. I hope your listeners have found something interesting. I do like to talk about my research in my cells in the laboratory, but it's also very nice to be able to get out and really see where maybe the potential impacts of your research may be. So it's an extra motivator to go back into the lab and get some more experiments done.
Well, this is very, it will be thought of as very nicely, and we get from there. Thank you for the recording.